Hello, Dr. Dan Guerra here from Authentic Biochemistry Studios in the Inland Pacific Northwest. Today, of course, is the 8th day of March, and it's the year 2021. Now, I thought I would do a video lecture today rather than a podcast audio because sometimes it's easier to use slides that you can see rather than just hear me giving a lecture and not having some kind of representation near it. So that's why we're doing it this way. So, from the beginning, this particular lecture is going to be called Excitatotoxicity, Neurotransmission and Amino Acid Metabolism and Age-Associated Neurodegeneration. This is going to be lecture 1, because I'm sure there's going to be more than one, because there's a lot of material I want to do. Today, as I said, is the 8th of March. My name is Dr. Dan Guerra, and there's really no reason for me to do any more introductions, is there? We're doing this because I have nothing better to do, which means I love doing it. Now, the paper we're going to spend more time on than any other paper eventually is this one. It's the Journal of Pharmacological Sciences. It came out in November just of last year, so it's 4 months old, and there's the page numbers. So put myself back up here. Now, what this paper is going to do, I broke down in a summary way, and I also had a lot of new materials on this first page. There's a poise, of course, between excitatory and inhibitory neuronal transmission. The bulk of central nervous system excitation, including that excitation associated with cognitive, motor, sensory, and autonomic activities, actually runs through an amino acid that we are all very familiar with, which I've been talking about in great length in the podcast lectures, and that's L-glutamate. It's one of the most common non-essential amino acids, right? And we've talked about its interconversion. We talked about its control via acylation of many of the enzymes involved in the regulation of carbon flowing in and out of the glutamate-glutamine cycle. Remember that? The TCA cycle and all that was all covered in the last 3 or 4 lectures on the podcast. So that's where this is coming from, and that's why we're continuing it now by including first some physiology and then some pathophysiology. So there's a balanced and regenerative glutamate homeostasis. It's essential to prevent many of the possible neuropathological outcomes that can happen, including neurodegeneration. And classical epilepsy also is linked to this particular neurotransmitter, L-glutamate, okay? And all the pathophysiology that you might imagine. Multiple sclerosis, which is a central nervous system disorder, right? It's a demyelinating disease. Of course, Alzheimer's, Huntington's disease, amyotrophic lateral sclerosis, or Lou Gehrig's disease, and of course, Parkinson's. Now, in AD, prodromal synaptic dysfunction is associated with an increased oligomeric amyloid-beta peptide, which we all have heard many times, and that is linked to an NMDA receptor response, and particularly an NMDA receptor dependent synaptic dysfunction. And this is going to lead into spine elimination as well, dendritic spine elimination. Now, although AD biomarkers classically since the 1990s, maybe even the 80s, have included extracellular amyloid plaques composed of the amyloid-beta protein, or the A-beta peptide, and indeed intraneuronal neurofibrillary tangles, NFTs, associated with abnormally hyperphosphorylated tau protein, that's the tangles. You also get necrotic or apoptotic neuronal and synaptic damage in AD. And the pathophysiology that really is linked to it is more that neurodegeneration than it is the plaques, the presence of the A-beta protein, except perhaps most significantly in its oligomeric form, and only certain of those, and certainly also the tangles. But the number one thing is neurodegeneration. That seems to be linked to one of the major psychological aspects of Alzheimer's disease, particularly the early stages of it, which is memory loss. Okay. So, since the NMDA receptor is a ligand of glutamate, okay, which is the primary excitatory neurotransmitter in the human brain, right after all, much research has been focused on that particular functional paradigm, glutamate binding through its NMDA receptor. Okay. Now, the NMDA receptor subtypes require the coincidental presynaptic glutamate release and the strong postsynaptic membrane depolarization. And that has to relieve what's known as the magnesium block, a divalent cation, of the channel. You'll see this when we go through the lecture. So NMDARs, the receptors, are permeable, of course, to sodium and potassium. They have pretty high permeability to calcium divalent, which acts as the second messenger, actually, to modify the synapses downstream from the initial burst. NMDARs are linked to the mechanism underlying brain plasticity, which is learning, which is associated with learning, and epigenetic phenomena. And that's being functional for mediating specific patterns of long-term changes in synaptic structure and the function associated with higher cognitive functions. Now, that latter part there was from a paper published a few years back, Frontiers in Neuroscience, February of 2019. So it's 2 years exactly, 2 years and a month, I guess. Not quite. So let's do some basic neuroscience. So you have an excitatory neurotransmitter, and it binds to its receptor. What happens is there's an influx of sodium, which depolarizes the membrane, and then increases the likelihood of generating, it's not guaranteed, of an action potential. An inhibitory neurotransmitter, trafficking chloride anions, you get an influx of chloride, that keeps the membrane polarized, okay? Negative on the inside, positive on the outside, in terms of polarity. That decreases the likelihood of generating action potential. That's why it's called an inhibitory neurotransmitter. And that's why the one that's using glutamate, for example, using the sodium importation is going to be called an excitatory, because it's going to change the polarity of the membrane, because notice the negative internal part of that membrane is going to be altered because you're pumping in cation. Right? See how that works? Okay. And everything is about the membrane. So we can tell you real briefly about the types of neurotransmitters. There's excitatory, inhibitory, and then there's some neurotransmitters that function at both levels. The excitatory, primarily glutamate, aspartate, and even nitric oxide. And the inhibitory, glycine, gamma-aminobutyric acid, serotonin, dopamine. And both excitatory and inhibitory neurotransmission can be affected by acetylcholine and norepinephrine. Let's talk a little bit about this whole membrane potential. The permeability of ions across the bilayer sets up a charge distribution because of membrane transport proteins, right? The membrane is a bilayer often. It's not necessarily a bilayer, but often is. And across that bilayer, you have intramembranous proteins, which can have a lot of alpha helical secondary structure, allowing them to be ribboned through that membrane. Right? And that's what we're talking about here, the classical membrane proteins. So the resulting charge density across the membrane will generate electrochemical difference. And that can be described by delta psi equals psi inside minus psi outside. That is basically the membrane potential. That's what the delta psi is. Now, the driving force for non-mediated molecular transport through any medium is A, whatever A happens to be, for example, a neurotransmitter or an ion, A is electrochemical potential gradient given by what's known as the Nernst-Planck equation, which is shown there. JA equals negative A, U sub A, change in GA divided by dx. So now let's define all our terms quickly. J sub A is the flux or rate of passage, okay, through a unit area of substance we're calling A. Okay. X is the distance. Delta G sub A divided by dx is the electrochemical potential gradient of substance A. The event of that crossing is that ratio. And U sub A is the measure of mobility of whatever that substance is. And that's going to be indicated and almost dictated by its solubility in either lipid or aqueous, as we shall see. So to flesh out the membrane potential in more detail, I think I did mention this quickly on the audio lecture, but here you go, just you can see it. You look at the Nernst equation here, where the m to z is the ion of charge z. Okay, there's your delta psi. It's going to be equivalent to RT divided by ZF, natural log of m to z outside divided by m to z inside. So, of course, the delta psi is the membrane potential in volts, and Z is the charge of the ion. Remember, I was talking about divalent, monovalent, cations, and anions. The F is the Faraday constant. R equals 8.3, you see there, and that's joule per degree Kelvin mole. And basically, that's the universal gas constant. So you can derive also something called the Goldman equation, which is more important in biology. And that takes into account multiple ions, that's why it's important, and different permeabilities of the ions, which is more in the natural physiological state. So you can see that equation there. It's going to have a couple of new terms. First of all, it's going to sum all cations involved, right? And so you have sum of all cations involved, sum of all anions involved, and you also have these P's, and the P's are the relative permeabilities to cations and anions involved, okay? Other than that, the Goldman equation pretty much fits the same exact series of quotients that does the Nernst equation. Okay. So now we have to get into Fick's law. Okay. Fick's law, first law of diffusion, is the following. Any substance diffuses across a membrane in the direction it collapses the concentration gradient. So that's the delta A is the substance, over the distance, that's delta X, right? And that's a rate dependent upon the magnitude of the gradient. The Nernst-Planck will reduce to JA equals negative DA delta A divided by delta X, where DA equals RTUA, which is the diffusion coefficient of substance A inside the membrane. Okay. And a new term, that's PA equals DAX, divided by X, I should say. That's the permeability coefficient for substance A. So the permeability coefficient is important because it's indicative of the solid's tendency to partition from aqueous into lipid. Okay. And lipid, of course, is hydrophobic, and that's going to be the center of the membrane. And why it's going to be hydrophobic is because of the fatty acyl chains. Right? They're always going to be in the interior in a bilayer. Okay? And that'll carry, that will vary according to the ratio of the solid solubility in acyl lipid to that in water. Okay? Makes sense. The ratio of the solid solubility in aqueous to that in a hydrophobic solvent, because that's what the lipid moiety is like in the middle of the membrane. That is known as the partitioning coefficient, because partitioning from a polar solvent into a non-polar solvent. Right? So you're partitioning, of course. Now, this can be described by this graph here. We've got flux on the x-axis, and you've got concentration of substance A outside minus concentration of substance A inside. That's the x-axis. The y-axis is the flux in unit area, JA. So you can generate a couple of different descriptions here. First of all, here's your electrochemical potential of A, shown by this equation. Okay, it's just a rearrangement of the one we were looking at before. And we can also see that we can develop the slope of this line, which is going to give us the permeability coefficient, the P sub A. So permeability coefficient equals the diffusion coefficient divided, of course, by the thickness of the membrane, that X value. Right? So the diffusion coefficient is the velocity per unit time times the RT, right? The constant temperature. So R is the gas constant. Okay, remember that. There it is again. Joule, degree Kelvin, mole, reciprocal mole, and temperatures is just in degrees Kelvin. Okay, so that's how you get the slope of the line. And again, this is describing the electrochemical potential and also the permeability coefficient and diffusion coefficient of substance, here we're calling A, across a given membrane. Okay. Now, you can take a look at some of these compounds, look at the permeability, and now you'll learn the relationship between permeability coefficient. Here it's going to have units of centimeter, reciprocal seconds, and then this oil water partition coefficient, which is going to go from 0.0001 to 0.01, so 2 orders of mag. Now look at the kind of compounds we're talking about. Urea, which I'm glad this is here because we talked about the Urea Cycle last time. There's urea, one of my favorite compounds. I don't know why. I like its symmetry, I think. You can see that its oil-water partition coefficient is really low, and its permeability coefficient is really low. And then you start getting more hydrophobic compounds, ethylene glycol, ethylurea, acetamide, formamide, succinimide, diethyl urea, propionamide, and N-butyramide. You can see it's getting more hydrophobic. Highly hydrophobic compound way up here in the upper right. It's going to have a very high oil-water partition coefficient. And it's going to have a 2 orders of magnitude higher permeability coefficient. Okay, so now you see the aspect of hydrophobicity of substance A, which we just showed you first in the graph and then the equation. And now you see it graphed. Okay, so you get an idea of what will cross easily, right? Move faster, have a higher permeability coefficient. It's going to be something that's hydrophobic. Something that's quite hydrophilic, like urea down here, not going to move very fast. Okay? And therefore, it will demonstrate with a very low oil-water partition coefficient. Okay, so now you understand how the parameters are associated with one another from a simple graph. This shows you just the flux of glucose into human erythrocytes at 5 degrees C. You can see it's a hyperbolic relationship with the concentration of glucose on the outside increasing on x-axis. And then the movement of glucose, here's your J value, you see, flux, in millimoles per centimeter reciprocal seconds. And this is times 10 to the 6th. Here's the maximum it can do, which is about 1 times 10 to the minus 6th, right? And never quite reaches that, it becomes asymptotic. And you can draw down here a half maximal. And if you draw down the half maximal and take it to the x-axis, that will give you the Km of that movement of that substance through the membrane. Okay, the Km, remember, means half maximal, but really has more to do with the concentration of the substance that will give you half maximal migration. So that's why we're looking at the Km being evaluated as a concentration here of glucose. Right? So the flux of glucose to the human erythrocyte functions this way. In a hyperbolic manner, right? It increases as you increase the amount of glucose. Remember, glucose is quite water soluble. Okay, that's why you have these relatively high values, right? Okay. So going back now to the cartoon, you start off with A coming. A is outside, right? And you're going to have it going from outside. It's going to be associated with a component of the membrane protein. It's going to be transported to the inside. and then A is going to be lost in dissociation, then the transporter will move back across and recover to come back outside to pick up a new concentration of substance A. This is a very simple diagram showing you, which is far from simple when you think about the complicated effort that does occur across the membrane to run solutes through, right? But this is basically showing you in diagram form what we're talking about, right? Okay. So what mechanisms you got? You can have a carrier ionophore where the ligand or the transported solute can bind into the protein. The green horseshoe here is the protein. The red ball is the solute, and it can move by flipping from the outside of the membrane to the inside of the membrane, thus releasing its contents. That's a carrier ionophore. Or you have channel-forming ionophores where the substance will move through a protein channel. And then the amino acids that make up that barrel inside that barrel are going to be specific to be able to mobilize that particular solute across the membrane, dealing with its polarity and, in some sense, its molecular size, because it acts as a molecular sieving process as well. Okay. So now that's very basic. Let's go now into action potentials because we're talking about neurotransmission. The resting action potential is at negative 70 millivolt. Okay. Shown down here. This is time in milliseconds on the x-axis, and this is going to be the membrane potential in millivolts on the y-axis. So you get a stimulus. So from the resting potential at minus 70, it's triggered when the membrane potential reaches a threshold of about minus 55 after the stimulus. Okay. If the gradient potential change exceeds that the threshold, you actually get this action potential firing. Okay, and that's the threshold it has to reach. It has to go from minus 70 resting to at least minus 55. This is millivolts across the membrane. Okay. So then what you get is a very rapid depolarization, and that's a change from the minus 70, which is what you started with, shooting all the way up to plus 30 millivolt. Okay. And then you get a also equally rapid repolarizing phase, which is a reversal of the polarization. That's what that means. When you say depolarizing, repolarizing, it's just reversing the polarity. And then that's going to go from the plus 30 millivolts back finally to the minus 70. But you're going to have a little bit of an overshoot here. This is called the after hyperpolarizing phase. And then it will meet back up to the minus 7 resting membrane potential. So the action potential here that I'm showing you is describing a nerve impulse. It takes place in two stages. You've got a depolarizing phase, which is more positive, shown here, up to plus 30 millivolt across the membrane. And then a repolarizing phase, which of course is going to be more negative, right? Because it's just a reversal of polarization. It's going to go back down ultimately to the resting potential, which is out here, minus 70. Okay, that's followed by this hyperpolarizing phase. You see here, is your hyperpolarizing phase, or a refractory period. That's the period under this graph here, in which no substance A can be generated. So no potential can be generated, no action potential can be generated because substance A can no longer move once it's in this area down here. Okay. All right. So said again, at rest, typical values for membrane potential range from about minus 40 to about minus 90. Passing a positive electrical current into the cell will depolarize. That is, it will make the membrane potential less negative. In response to a small depolarizing stimulus, the neuron's response is small as well. In response to a larger stimuli above that threshold level of minus 55, the response is fundamentally different. The membrane potential quickly rises to a value well above zero. We showed you go up to plus 30. And then it falls over the course of 1 to 5 milliseconds. This all happens very rapidly, back down to the resting value after, remember, that lag phase period. Now, often, the falling phase of the action potential undershoots the resting potential temporarily. Okay, that's what I showed you. That's actually turns out to be pretty significant. The action potential is said to be an all or nothing response because it occurs only if and only if sufficiently large depolarizing stimuli, and because its form is largely independent of the stimulus for the supra-threshold stimuli, right, going beyond that minus 55. Right? So in some neurons, a single action potential can be induced by the offset of a hyperpolarizing stimulus. That phenomenon is called anodal break excitation or rebound spiking in neuroelectrophysiology. So the nerve impulses, of course, are propagated by what are known as action potentials. So here we get more into the detail. Voltage-gated channels, the stimulation of a neuron causes sodium channels to open. So the sodium ions spontaneously flow into the cell. That's how we started off this lecture. The consequent local increase in membrane potential induces a neighboring voltage-gated sodium channel to open. The resulting local depolarization of the membrane induces nearby voltage-gated potassium channels now to open, to respond to it. That allows potassium ions to spontaneously flow out of the cell while sodium is moving in. And that's the process of repolarization. Right? That's what you saw. However, well before the distribution of sodium potassium ions, which are both monovalent cations, move across the membrane is equilibrating, and the sodium-potassium channels will spontaneously close once the membrane equilibrates, given the relative concentration of sodium that's moved across and potassium that's moved across in the opposite direction. Yet, even though this occurs, because depolarization of a membrane segment induces the voltage-gated sodium channel in a neighboring membrane segment to open, which induces their neighboring voltage-gated sodium channels to open, etc., you get a wave of transient changes in the membrane potential, moving down the axon, okay? And that's called the action potential because it's traveling along the length of the nerve cell, right? And that can be over a meter in length in human neuronal transmission. So the action potential, which travels about 10 meters per second, propagates in one direction only because after the ion channels have spontaneously closed again, right, they resist reopening until the membrane potential is regained to minus 70 millivolts, the resting potential, which takes a few milliseconds to compensate and regenerate. Right? Okay. So as an action potential is propagated along the length of the nerve cell, it is continuously renewed so that its signal strength remains constant. This is essential. Otherwise, you'd have start, stop, start, stop, start, stop. And neurotransmission would act that same way, thus corrupting whatever the stimulus is and whatever the presentation is. So the reception will no longer give you a perception of space-time and whatever's changing within space-time according to your sensoria, right? So that can't happen, right? So it's got to remain constant. In contrast, when you think about just a normal electrical impulse down a wire, copper wire, it dissipates as a consequence of resistive and capacitive effects, right? Whether or not there's some discharge of the electron moving through the wire or there's an enhancement of it. Right? It doesn't happen in this case. Nevertheless, the relative ion imbalance responsible for the resting potential is relatively small. It's only minus 70 millivolt. And only a tiny fraction of the nerve cell's overall sodium-potassium gradient, which is capable of maintaining. So that whole gradient is discharged by a single nerve impulse. Only one potassium ion per 3,000 to 300,000 in the cytoplasm is exchanged for the extracellular sodium ion. The neuron then will transmit a neural impulse every few milliseconds in perpetuity until the stimulus obviously gets removed, right? So this capacity to fire rapidly is an essential feature of neuronal communication. Since action potentials all have the same amplitude, and I showed you that amplitude from minus 70 to 55 to about plus 30, and then back down, the magnitude of stimulus is conveyed by the rate of the firing or the stimulus. Right? So that's how charge distribution is affected by the intensity of the transmission. Because you're still within a very fine window of the polarization shift within the membrane. So it has to do with the input of the stimulus initially, the overall effect of the impulse on whether or not the membrane is receiving a powerful signal or a weaker one, as long as you get back past the minus 55 barrier, that is. So this is shown here. These are myelinated sheaths. So myelin is sphingomyelin. That's a lipid that covers certain nerve bundles in the central nervous system. They're not all myelinated, but some are. The myelination allows for even more rapid movement of the action potential via this particular mechanism. So I'll summarize it first. Initially, the threshold stimulus creates an action potential, like I just showed you. As the nerve fiber depolarizes, it triggers an increase in the permeability of the ligand-gated sodium channel, right here, okay? That causes sodium channels to open, right? So now they're opening, like I showed you in the first slide. And sodium ions go into the nerve cell, and then reverse polarity of that membrane because positively charged ions are flowing in. The action potential then is going to be conducted along the length of the fiber, see here, by inducing the next adjacent space, which is available, right, to open another voltage-gated sodium channel. So you have the voltage-gated sodium channels in between where the myelin is. So the myelin isn't uniformly covering the neuron. It is altered with sphingomyelin, and then it stops. Sphingomyelin, and then it stops. That allows you then to propagate the neural signal at a higher rate because you have an insulation of the process. And you don't have to fire along a continuum of a series of sodium voltage-gated channels. You only have to hop to the next one. That's what it's called, saltation or hopping. Right? So that's exactly how it works. So sodium is entering the cell due to these voltage-gated sodium ion channels, right? Opening up, and meanwhile, to the left of it, the potassium is exiting. Okay, I'm not showing you that here, but that's what's occurring. So as the potassium ions move out, you get that repolarization effect. And so you start tuning down, dissipating the neural signal because it's moving down this fiber. So then after this occurs, the positive charge will move to the adjacent region of the membrane. See, now you've got positive here. You have the positive region here. Now you've moved that, you've transmitted that concentration gradient, that permeability coefficient is now leading down this action potential to the next, what we call these nodes of Ranvier. Those are the unmyelinated regions of a myelinated nerve fiber in the central nervous system. The node is. Positive charge will move to the adjacent region of the membrane. As the charge moves, it will then depolarize the membrane, just like we saw, okay? If it depolarizes sufficiently, that is, if the nerve impulse is strong enough, voltage-dependent sodium channel in the adjacent region will respond in kind. And it will be opened up, and a new action potential will then be initiated. Okay, it'll start all over again, but now we're moving the potential down the nerve fiber. And we're enhancing its movement by having it insulated by the sphingolipid known as sphingomyelin. Right? Okay. So then you get a new action potential initiated, and that charge distribution will then spread to the next region and on, initiating new action potentials as it moves through, changing the polarity at the node, and then moving on and changing the polarity, and moving on and changing the polarity, all the time dealing with sodium influx through that node. And the myelin is allowing that charge distribution to be saltated or to bounce from one section of the membrane to the other without dissipating, because it isn't a naked nerve fiber, it is myelinated. But it's not completely myelinated. If it were, how would you get the sodium ions across? Because sphingomyelin is a very hydrophobic sphingolipid. Right? So that's usually not explained in biochemistry books. That's why you have to have naked, unmyelinated regions. Those are called the nodes of Ranvier. Otherwise, you would get none of this saltation process, and you wouldn't get any neurotransmission either. But what the myelin does is enhance the rate of movement of that gradient, right, down that membrane. Okay, I think I explained it pretty well. We just got one more thing to clean up here. Propagation of the action potential in myelinated fibers is illustrated here, and I showed you last time as well, starting with the action potential at a node on the left, okay? In the absence of myelin, the action potential would propagate actively through the simple mechanisms we already showed you in the very first slides. However, see if I've already explained this, now I wrote it again so it would hit home to you. The myelin occludes all the voltage-dependent sodium channels. There's nothing there. There's no sodium channel where the myelin is, you see. So the action potential cannot propagate actively in that region. In fact, myelinated axons do not even have sodium channels in the internodal region. Rather, the potential change produced by the action potential at one node spreads the internodal region along the axon passively, just as the temperature would spread along a long metal rod. So it doesn't even need to have channels in those non-myelinated regions. You'll still get a propagation of the neural transmission, of the action potential, because now it is free to move through, because it is an unbounded, unmyelinated portion where you're going to be able to get the polarization to occur. The whole process is driven by the change in polarization. Now, we can stop here and just talk a little bit about disease, because we are authentic biochemistry. We talk a lot about pathophysiology. Myelinated sheaths and multiple sclerosis. I told you many of the diseases that we talk about in aging, central nervous system, have to do with glutamate and the NMDA receptor. We're getting to there, and now I'm stopping here and talk about MS. Now, MS isn't necessarily a disease of aging. Very young people very sadly can get it, and it's devastating for them. But this is why, this is how it happens. Myelinated sheaths interrupted with nodes of Ranvier, we just saw. Here, the axon is in contact with the extracellular medium at that nodal interval. I see that as a space-time because it's always moving through the space of the neuron as well as the time flux of the action potential along that nerve fiber. So it's a space-time event. So the sodium channels are located at high density here, as I just showed you, at that unmyelinated region, and the action potential saltates. Again, that's saltare, which is Latin for jumps. Okay? So neural transmission obtains due to the ionic current via the passive conductance where myelin is. Okay? Because remember that myelination then is a protected region, right? No dissipation of the electron-driven force, right? Of the electrochemical potential. So this is faster than a continuous propagation would be of an action potential, that is, but it's also open to rapid dissipation because you have this insulation. And that's like electrons moving through an uninsulated copper wire, you see. Therefore, the nodes amplify the signal to maintain the intensity of the electrical stimulation, and the myelin sheath creates the environment for this to occur. Without myelin, which is multiple sclerosis, is a degradation, an autoimmune disease which causes the myelin to be degraded. Without myelin, the electrical impulse becomes attenuated, that means stopped. Ion leakage starts occurring, and the impulse doesn't capacitate to the next node. That is basically multiple sclerosis pathophysiology. Yeah. So now we're going to look at this cartoon and think about what are the names of these proteins which are carrying out the action potential and driving neurotransmission. Well, we have two types of glutamate receptors. We have the ionotropic and the metabotropic. The ionotropic include ones that can push sodium and calcium, see here, and they include the AMP-A, the GLUT receptors 1 through 4, the kainate receptors, named after this compound which acts as an agonist for glutamate receptors, and those kainates are also known as glutamate receptors 5 through 7, or K1 through K2. So you've got 1 through 4, 5 through 7. Then you have the classical NMDA receptors, right? N-methyl D-aspartate, and you have NR1 and NR2A through D. Okay. Now these are the ones that are going to utilize that magnesium I told you about, associated with that excitotoxicity that we started the lecture with. Right? Of course. See, I'm always trying to bring this back with you. Now that's going to generate ion currents. This kind of receptor, the ionotropic glutamate receptors, are going to generate ionic currents. Now the metabotropic still uses glutamate. This is glutamate. It binds to its receptor. And here you have the mGlu1s through mGlu8. So there's 8 isoforms. And they're associated with the trimeric G protein, the alpha beta gamma protein. And the alpha portion will go to another protein embedded in the membrane, and also will function at the effector molecule level. So this will be a metabolic transmission of the neural impulse, because it's going to involve secondary messengers like cyclic AMP, phosphatidylenosyl trisphosphate, compounds like that. And that's where they're going to then meet out their role in neurotransmission by dealing with the secondary messenger synthesis and then activation of protein kinases, for example. Whereas these other receptors here, still using glutamate, they're all dealing just directly with electrochemistry. Right? They don't necessarily have any secondary messengers. Okay. So, pharmacotherapy for MDD, now that's major depressive disorder, still predominantly at a monoamine axis, despite ambiguous efficacy and large placebo effect of the serotonin and norepinephrine reabsorption uptake inhibitors, right? New therapy has been targeting, though, the glutamate neurotransmission. That's why I bring it up right now. The central excitatory neurotransmitter in CNS, of course, is glutamate. I've said the L-glutamate. Well, GABA is the major inhibitory amino acid neurotransmitter. So you've got gamma-amine butyric acid is the inhibitory, and glutamate, which is the excitatory. Okay. Glutamate seems to be linked to something about glutamate transmission to major depressive disorder. So the N-methyl D-aspartate receptor, we just looked at it, binds to and is activated by glutamate, but also by glycine and glutamine. Remember the glutamate cycle I showed you? Remember that? Remember getting rid of the amino group and converting glutamine to glutamate, and possibly glutamate being converted alpha ketoglutarate to generate enough energy running through the TCA cycle, NADH, FADH2 production to make ATP. You need a lot of energy to do neurotransmission. So one of the things that happens, you get the glutamate moving through, or the glutamine moving through, but you utilize that carbon, and you, in the process of oxidizing that carbon in the TCA cycle, you reduce those nucleotides, and then you reoxidize them in the electron transport chain in the mitochondria of these neurons to be able to drive enough ATP synthesis to synthesize the neurotransmitters, allow them to generate synapses, and allow the synapses to refold and reform, at the same time carry out all the energy necessary for the action potentials to function. So there's an antagonist of the NMDA receptor, which you may have heard about. It's called ketamine, and it has efficacy in remitting some major depressive disorders. That's interesting, but it's something that's being looked at, and there are psychiatrists that do prescribe ketamine to some patients. And it is a drug that seems to work, but it is not 100% efficacious because no drugs are. So gamma-butyric acid binds to its own class of receptors, as you might gather. So it does not directly inhibit the NMDA receptor or any other glutamatergic loci, the ones we just looked at, the inotropics versus the metabotropics. So that's all important to know. So this is going to show you now. Here's glutamate moving through the NMDA receptor, and there's magnesium bound to the NMDA receptor. There's sodium moving through the AMPA receptor. So molecules of glutamate bind to the recognition sites of the receptor, okay, both to the NMDA or the AMPA, as well as the AMPA, and the ionotropic AMPA receptor admits sodium ions when activated, and that results in a moderate local depolarization. Okay. Now notice that you've got magnesium blocking here, right? Now watch. What occurs during that depolarization is you get a dislodging of the magnesium ion because of that. Magnesium ion was preventing the NMDA receptor from greeting the glutamate signaling, right? In that process of the ionotropic action potential being generated. So it dislodges the magnesium, which is blocking the NMDA receptor. So large quantities of calcium ions, which has got the same valency, different Stokes radius. Of course, calcium's a really important secondary messenger, that's right. Allows calcium ions to enter the neuron through the NMDA receptor, the calcium, which are called the calcium channels now. And so the NMDA receptor is thus both ligand and voltage gated. Right? So both things are occurring, voltage gated and ligand specific. Okay, and that's part of the metabolic machinery of the cell. Finally, you see here this magnesium comes back, binds to its NMDA receptor, but glutamate can work through all the A receptors without any inhibition and driving that sodium channel redox that we just talked about. So that results in the addition of more AMPA receptors. Once you've done this initial process with the NMDA, you recruit more AMPA receptors to the surface of this synapse, right? And so with more AMPA to the postsynaptic membrane, because this is going to be post, because the presynaptic membrane is sending the glutamate out, because that's the neurotransmitter, right? It's going to place more AMPA receptors there, so you get a stronger signal. You get a more stronger stimulation postsynaptically after the glutamate transport through the synaptic space, right? Coming from the presynaptic neuron now to the postsynaptic neuron, inducing more AMPA receptors to that postsynaptic neuronal membrane. That's what's happened. The synapse thus has been strengthened, they call it, and they will respond more rapidly and more strongly to all future releases of glutamate coming from the presynaptic neuron. Now, something to talk about in terms of protein modification. Protein modification can trigger many things, including neurotransmission phenomena. We talk a lot about protein modification, glycosylation, acylation, prenylation, 3 we've talked a lot about, right? Altered expression of receptor isoforms via Sirtuin. Yeah, remember the deacylases, deacetylases are back. Told you I would bring them back in this lecture, and this is where I'm doing it. So Sirtuin 1 inhibition increases the expression of the glutamate A2 receptor, glutamate A3, and the glutamate K5, that's the kainite, right, or kainate, and a metabotropic glutamate receptor. This was all published in Scientific Reporter 2016. That's all induced when you get Sirt1 inhibition. How can you inhibit Sirt1? By removing NAD+. Altered activity of the metabolomic enzymes via covalent modification also includes this acetylation-deacetylation cycle. That was published way back in Cell in 2006. We talked about that paper in the audio lecture. Sirt4, that's the mitochondrial sirtuin, inhibits, remember, glutamate dehydrogenase, right? Because Sirt4 will deacylate glutamate dehydrogenase, and that opposes the effects of calorie restriction in pancreatic beta cells, which would otherwise be secreting insulin, right? Remember, we talked about this in the audio lecture just last time. Also, recreational drugs like methamphetamine, bad drugs, can alter glutamatergic receptor gene expression via an epigenetic modification of the histone H4 hypoacetylation. And methamphetamine induced decreased striatal glutamate receptor expression. And that was first published in Biological Psychiatry some 7 years ago. So there's a lot of pleiotropic effects of drugs altering neurotransmission that aren't normally considered when you're thinking just merely of an addiction to a drug, right? But there's a lot of membrane effects with alcohol too that I talk a lot about during the holidays when people drink a lot. And I won't give that up this time. I will talk about how alcohol has very similar disruption of the membrane, but it's less specific than things like methamphetamine, cocaine, drugs like that. So remember that Sirtuin deacetylation and control over protein transcription. This is just a reminder to you. So you've got an acetylated protein, right, on a lysine residue, right? And Sirtuin will deacetylate it, right? The same time, it will take NAD and convert it to NAM, nicotinamide adenine mononucleotide, right? That NMAM has to be then processed, has to be reacted to go back to making NMN, or nicotinamide mononucleotide, right? So that has to happen by this transferase enzyme. Once you get that, it will then you can synthesize NAD with the enzyme NMNAT, the transferase, right? Now you produced the dinucleotide, and now you're back to being able to be a functional sirtuin because you need NAD. So there's a whole cycle here now. You also, an intermediate, can make 2-O-acetyl ADP ribose. Remember the ADP ribosylation reactions that sirtuins do. That's an intermediate in the cycling of the NAD through the NAM to the NMN and back to the NAD. That's 2-O-acetyl ADP ribose can be generated. All right. So this whole acetylation process itself requires this transformation of the NAD moiety. And that in itself requires all of these enzymes. And if these enzymes are deficient or otherwise altered even by sirtuin activity, which some of them are, then you're going to have a corruption in neurotransmission. When you need this sirtuin to mediate responses in the membrane, to alter the flux through the channels, as well as recycling the glutamate, alpha-ketoglutarate, and glutamine, because of the acylation, deacylation of the enzymes responsible for those cycles. Okay. All right. Now we're back to this paper, the Journal of Pharmacological Sciences, again published just about 3 months ago. At the excitatory synapse, glutamate is continuously released from the presynaptic neuron and recaptured through the well-described glutamate-glutamine cycle that I talked about in audio lecture. After the glutamate is released by neurons into the synaptic cleft, it can bind to both the ionotropic and metabotropic receptors. And either way, it's going to transmit an excitatory signal to a sufficient amount of glutamate. Subsequent to the glutamate levels rising and getting up to about 1 millimolar at the synaptic cleft, in less than 10 milliseconds, it's rapidly removed from the cleft and its concentration returns to nanomolar levels. So that goes from nanomolar to millimolar, right? So that is what? From 10 to the minus 9th to 10 to the minus 3rd. Very rapidly in less than 10 milliseconds. So you have those, that means you have very high affinity, high responding transporters. And of course, you get those transporters in both cell types, in glial cells like astrocytes and oligodendrocytes, as well as, of course, in neurons. And we talked about how we cycle that carbon from the oligodendrocyte, right, to the neuron, and then from the neuron back to the oligodendrocyte. The glial cells are actively participating in neural transmission. Right? Okay. So in astrocytes, the enzyme glutamine synthetase, which synthesizes glutamine from glutamate and ammonium ion, said here, converts glutamate into inert glutamine, which in turn can be properly released, taken up by the neurons. So this is happening in the astrocytes, which are adjacent to the neurons. So you make glutamine, which is not going to be processed in the astrocyte, but it can be released and taken up by the neuron as a precursor now for glutamate. And also you're going to release ammonium, so you need that CPS1, right? That very first carbamyl phosphate synthase 1 to remove the ammonium. And that's why you get the erotic acid synthesis. Remember that? We talked about that in Authentic Biochemistry lecture. Okay. So the overexpression of SIRT4, remember this is a deacetylase in the mitochondria of these cells, will decrease glutamine synthetase levels. When that occurs, you inhibit glutamine formation, obviously, which would otherwise be converted to glutamate in the neuron. So you're lowering the dose of glutamate entering the neuronal processing, the synaptic action potential we just talked about for all those receptors, the inotropic and the metabotropic. Okay. So SIRT4 then is doing what? It's preventing the potential for excitotoxicity, because remember, glutamate is an excitatory neurotransmitter. So SIRT4 is there to ablate the potential for excitotoxicity, which I told you is prodromal to diseases like Alzheimer's and Parkinson's. Right? That's right. So SIRT4 prevents excitotoxicity by upregulating glutamate metabolism, by reducing glutamine synthase expression and upregulating those other two proteins we've been discussing, right? The glutamate transporter and glutamate dehydrogenase activity. Right? So it works perfectly to detoxify the potentially dangerous excitotoxicological state of these neurons in the central nervous system in the aging brain. Now that last bit in red there came from Human Experimental Toxicology, published in July just of last year. So we take a look at this. This is the cycle, right? So you got the astrocyte sending glutamine. The glutamine is then converted to glutamate. Glutamate enters because of metabolism to alpha-ketoglutarate, right? Via the aminotransferase reactions, the transamination reactions. And so the TCA cycle is going to be functioning there because it's anaplerotic to bring glutamate back in from the alpha-ketoglutarate pool after the dehydrogenases slow down because of the buildup of NADH. Right? So then the glutamate transporter will then move through this glutamate transporter here, right? This is the process of moving now from the neuron back to the astrocyte. Then you can have glutamate going through the same metabolism, for example, glutamate dehydrogenase to the TCA cycle. You can also get TCA cycle intermediates moving all the way through to make lactic acid, right? And glycolysis can feed back into the TCA cycle as well. All right. This is where glucose is being used now to drive this whole system to get lactate to be metabolized in the TCA cycle, to make glutamate, and then glutamate can move back through the process of the glutamate transmitter. So you have glutamate, glutamine transmitters going from astrocyte to neuron. And neuron's where you're going to get the X potential generated, right? So this is the glutamine, glutamate transporting cycle between those two, the glial cell and the neuronal cell. Now, branched-chain amino acid transaminases also function here because they will synthesize acetyl-CoA and acetoacetate, which can be used, the acetyl-CoA can be used to acetylate proteins, thus controlling the expression of some of these genes by generating euchromatin so that you make the neurotransmitters. Because now you've opened up euchromatin epigenetically via the deacetylation process of the histone lysines, so that you can then do messenger RNA transcription for the genes responsible for the synthesis of glutamine, glutamate, gene processing proteins, right? Carbon processing proteins from gene expression. All right. So, again, going back to this Journal of Pharmacological Sciences, November, neurons then can directly uptake glutamate, thereby, eo ipso, replenishing presynaptic glutamate concentrations, bypassing the glutamate-glutamine cycle altogether. Okay? And that's phenocopied. You get the same effect by SIRT4 because of what it does to glutamine synthetase. See? So glutamate uptake occurs via 5 specialized glutamate transporters. We talked about those. And those are defined as excitatory amino acid transporters, or EAATs. That's where that nomenclature came from that last slide. Excitatory amino acid transporters, or EATs. And there's EAT 1 and 2, and they're more common glutamate transporters. Both of those are the more common glutamate transporters in the central nervous system in humans. In more detail, the EAT1 isoform is mainly expressed on astrocytes, and the EAT2 is more responsible for 90% of the brain's glutamate reuptake, highly expressed on the astrocytes and on neurons in the plasma membrane. So EAT1 and EAT2 are present on oligodendrocytes, while microglia, which do not express glutamate transporter at a steady state, start to synthesize the transporters when they become reactive. Okay? Reactive means inducing a potential immune response. Now they're going to be able to take glutamate completely out of the equation because these are microglia. Microglia are going to use the glutamate to do what? Drive the biosynthesis of potentially pro-inflammatory cytokines, which can lead to neurodegeneration in the aging brain. Overall, therefore, the expression pattern of glutamate transporters in all those different glial cells, the astrocytes and the microglia, right, implies a crucial role for the maintenance of glutamate homeostasis to maintain a normal physiology in the central nervous system. This is why I brought this all together now, which is what we're doing. All right. And that puts it together here. Sorry for the fuzziness of this slide, but you can get hyperammonemia when you have too much of a buildup of ammonium because you get an accumulation of glutamine in the astrocyte. Okay? So hyperammonemia increases the conversion of glutamate to glutamine by the glutamine synthetase. Okay? And that will lead, the resulting increase in glutamine here will result in hyperosmolarity and mitochondrial dysfunction. That causes astrocytic, this is the astrocyte, this is the glial cell, this resulting in this pathophysiology, astrocytic swelling, and it'll impair the astrocyte's functioning in that cycle to the neuron. Here's the neuron, here's the presynaptic neuron that's supposed to be receiving the glutamine for conversion to glutamate to move to the postsynaptic neuron to generate the action potential to carry out the neurotransmission through the NMDA receptor, for example, or the AMPA receptor shown in the cartoon. So increased glutamine formation also decreases total brain glutamate stores because it's a source, right? Impairing the excitatory neurotransmission, which is mediated by the NMDA and the AMPA receptors. And there's also the kainate receptors that are not really shown here. And neuronal energy production is also going to be decreased because you're going to have less alpha-ketoglutarate production necessary for the amount of ATP needed to do all this work to move the neurotransmitters through the synaptic space from the presynaptic neuron to the post. That will deplete alpha-ketoglutarate and it'll further impair energy metabolism, of course, in the brain. So we're going to stop there. Right? And we're going to lead into then, that will lead us in subsequent video lecture I'm going to do. Sorry, it's getting so dark. I didn't realize I was supposed to modify the lighting. I guess my producer wasn't working. The producer's me, so that's why the image got dark, right? But it's okay. You don't need to see me. Hopefully that lecture was illuminating enough that you got the idea why we're doing all this. We're leading to the pathophysiology of Alzheimer's disease, for example. We even talked a little bit about multiple sclerosis, how these are all interlinked, the NMDA AMPA receptors, the kainate type receptors as well, metabotropic versus inotropic receptors, the action potentials that we fire through. And now we're starting to talk about how Sirtuin plays a role in that overall mechanism. And now that can lead to the production of pro-inflammatory cytokines, which can lead to neurodegeneration, which is a hallmark of what? The memory loss that we started way back with. Use your memory. Alzheimer's disease. Okay. So I'm going to stop now. This is Dr. Dan Guerra fading into the darkness. And we will return with a new video lecture soon enough. This is Dr. Dan Guerra from Authentic Biochemistry Studios saying bye.